And today is going to be a lot about how you assess gene expression changes in response to ligands. So from what you've learned about signal transduction so far, kind of the easy part is understanding that ligands and drugs bind receptors, and then all of these things happen within the cell, you know, engaging G proteins, different types of G proteins associated with different receptors, then activating effector enzymes, which generate second messenger molecules, kinase cascades, ultimately activating transcription factors in the nucleus. So one drug or one ligand can activate many types of genes, many different genes, perhaps hundreds of genes, and each cell has its signaling pathways set up differently. So each cell type is going to respond differently to a given drug. And so that's a lot of complexity. But one of the neat things with genetics is that there are great methods, even if you don't know all the intricacies of the signaling pathway, you can figure out which genes respond and don't respond to particular treatments, like adding drugs to cells or to human bodies. And so that's how, and actually recent advances in genetic technology make all this possible. So we're going to go through that today and see how all that works. So transcriptional changes mediated by signaling pathways. So how do we actually detect and analyze transcriptional responses globally? And what I'm going to do is go through a bit of history on this type of an effort to look at genome-wide responses to signals. And we're going to go through this kind of historically, but the reason we're doing it historically is that it's a great way for you to learn how next generation sequencing approaches work. So once the human genome was sequenced, we had in hand some very important information. We knew the list of all genes present in human cells. And there are right around 20,000 protein coding genes in human cells. Once we had all that sequence information, the first thing people did was they made microarrays. And a microarray is a solid support with a whole bunch of oligonucleotide sequences spotted onto that solid support in known locations. So you'd have all 20,000 genes. This is just showing a portion of a microarray, but all of a little sequence of maybe 25 bases or so, and each one uniquely corresponds to one gene. And it's all arrayed in a little grid pattern like this. And these are single-stranded pieces of DNA, one for each gene in the human genome. And then what people would do is you would get a cell type, say grown in a dish, and to one cell you would do nothing, it would be a control, and to the second dish you would add a drug, okay? And then you can take those cells and get all the mRNAs out of the cells, so control mRNAs, no drug, and all the mRNAs that are present after treatment with a drug. And you could take this pool of mRNAs and you do a little trick, you do reverse transcription, so you turn the mRNAs into cDNAs using the enzyme reverse transcriptase, which is a viral polymerase. And it does the magic trick of turning RNA back into DNA. So basically you take all of this messenger RNA, make a DNA copy of it, which is called a cDNA, complementary DNA, and then the control sample, you label all of those DNA molecules with a fluorescent molecule that makes them fluoresce green under UV light. And the experimental sample, we do the same thing, except that we would label all that material with a red fluorescent dye, okay? So this pool of green mRNAs turned into cDNAs corresponds to all the gene expression in the control cells, and the red labeled material in the form of nucleic acids corresponds to gene expression, mRNA content of these cells after treatment with a drug. And then you do this interesting thing. Since you have this array with oligonucleotides in known positions, each one corresponding to a particular gene, you could take this mixture of green DNA and red DNA and hybridize it to each of these spots, okay? And each of these spots actually contains thousands of molecules of exactly the same nucleotide. So you can get actually both colors to hybridize to a single spot because there are plenty of oligonucleotides there to accommodate them, okay? And basically, 4 things can happen. Here's a spot that has nothing hybridized to it, so that corresponds to a particular gene that doesn't make an mRNA in either the control or the experimental sample. Here is a spot that is green only. That means it's in the control sample, but not in the experiment sample. That means the drug turned that gene off. The drug treatment turned it off. Or you can have examples that are red only, and that's genes that got turned on by the drug treatment. Make sense? And the last thing that can happen is both hybridize, and that's a gene that is expressed in the control and the experimental sample, so it's on in either condition. So the interesting ones are the pure reds and the pure greens, because those ones responded to the drug treatment. If it's green only, that means that the drug treatment turned it off. If it's red only, the drug treatment turned it on. And then what you can do is scan all these results, put it under UV light, and it gets scanned and stored in a computer, and you can get the color and the intensity of each spot, and now you know what's happened to gene expression for all of the genes in the cell in response to treatment. Make sense? Okay, that worked pretty well. And here's what one of these microarrays looked like, and these are still in use today, but not as much. But you can see that here's the grid, and then there are like regions that are blown up here, and you can see all of these intensities. One of the interesting things is that when red and green fluoresce together, it makes a yellow signal. So all these yellow ones are expressed in both samples. But you see some red ones and some pure green ones, and those are the ones that are responsive. You see a lot of ones that have no signal emanating from them. Those are all genes that are turned off in either condition. Great, so you get all these 20,000 different assays for every single gene, one by one, all in one experiment. So pretty powerful method. And how do you then deal with this much information? Well, obviously there's a lot of computation involved, but what typically is done is the following. It gets turned into something called a heat map. And so a heat map is very interesting. It is a computer program that clusters genes into clusters of other genes that have similar expression patterns, okay? So each of these rows here is a different gene, and each of the columns, in this case, is a different cell sample, okay? And what you can see is that, and basically red means it was turned on very strongly, green means it's silent, it's turned off very strongly. And this is just a portion of the data, but it shows some of the genes that respond. So here are a set of 6 genes that are all green, means kind of typically silenced by the treatment in these cells. But in this cell type over here, they all get turned on, okay? So this is an example of looking at gene expression in a number of different cells. You could just as easily do an experiment where you look at total gene expression in one type of cell treated with many different drugs, okay? So you can use this any way you want to, basically. But it helps you make sense of the transcriptional responses in response to treatments. And the treatments can be using different drugs. You could use one drug and look at many different cell types. Anything you can imagine, really. You could use this to look at, a whole bunch of cancer samples in response to a drug, find out which patients are responsive and which ones are not. All sorts of things can be done with this technology. Okay, so that was kind of like technology that was around, the hot times for microarray analysis were probably the year 2000 to 2010, maybe something like that, or a little bit earlier. But next I want to backtrack and talk a little bit about DNA sequencing, because DNA sequencing, you need to know that in order to understand some of this next generation sequencing stuff. So here is how that's done. You need to understand the basic aspects of how nucleotides function during a DNA sequencing reaction. And you've all had this in like Biology 1107 or the equivalent, and also Dr. Neskevich mentioned this, I think. But the key thing here is that I want to draw your attention to the 5' carbon in deoxyribose and the 3' carbon, which typically has a hydroxyl group on it. So the 5' phosphate, triphosphate group is essential for polymerization, and so is the 3' hydroxyl group. And so this nucleotide here can be incorporated into DNA using base pairing rules, and then you can add the next one because it has a 3' hydroxyl. So what people did was, to sequence DNA, they made a nucleotide analog, just like we've seen for some drugs. And what did they do? This is a deoxynucleotide. This is a dideoxynucleotide, thymine in this case. And all they've done is turn that 3' hydroxyl into simply a hydrogen atom. So this nucleotide here can be incorporated into a growing strand using base pairing rules. It has to pair with A. But once it's incorporated, you can't extend that strand because there's no hydroxyl group on the 3' end. So it's a chain terminating nucleotide. And it's actually a dideoxynucleotide, because it's called dideoxy because there's no oxygen on the 2' or the 3' carbon. Okay, in the sugar moiety. So anyway, this one here base pairs with A, has a hydroxyl, and then you can add another nucleotide that will base pair with whatever that base is. But this one is chain terminating. Since there's just a hydrogen at the 3' position, DNA synthesis stops right there. And so what people did was figured, well, I'll do a sequencing reaction in a tube. We're sequencing a known piece of DNA, which is this bottom strand. And I'm going to make the reaction contain 99.9% normal nucleotides and 0.1% dideoxynucleotides. So 99.9% of the time, synthesis continues, but if this, 0.1% of the time, you add a dideoxy and that chain will terminate right there. So if you then do this sequencing reaction and you label it on the end, you actually do 4 sequencing reactions, one with dideoxy A, one with dideoxy G, one dideoxy C, one dideoxy T, and you can run them on a gel that separates them by size, and here's what it looks like. This is the dideoxy A column in electrophoresis, dideoxy C, dideoxy G, dideoxy T, and the smaller ones go further. So this is, as you read up this sequence, you can actually just read off the bases by looking where the band is, okay? And that is called Sanger sequencing. And it's done by radio labeling the ends of the DNA strand that you're synthesizing. And every single time you see a band, that means there's a termination event. So the resolving power of these gels is pretty phenomenal. It's 1 nucleotide. And so, for instance, right here you can see, 3 A's, and you read it from the bottom. So it's going 5' to 3' from the bottom. So this is like A, C, back to, over to T, back to A, and you can just read them from bottom to top. And so each one of those bands results from a chain terminating dideoxynucleotide that got incorporated. And people wanted to then make this more efficient. So this is a whole history of making things more efficient. Instead of running 4 lanes where the DNA fragments are radio labeled like this, they figured, why don't I do the whole reaction with all 4 dideoxys in one tube, okay, and then run it on the gel. But the trick there was they put a different fluorescent molecule on each of the sequencing reactions, and that distinguishes whether it stopped at a T, a C, or a G, and then you can run it all in one lane, similar to like before, but now instead of having 4 adjacent lanes, you can just have 1 lane, and the color indicates where the termination event occurred. So to do that trick, you actually need fluorescently labeled dideoxynucleotides, okay? So that was an improvement. You went from 4 to 1 lane. And then this is the technology that's kind of taken everything by storm. It's called next generation sequencing. And it makes use of that knowledge of DNA synthesis that I just went through, but it uses some different tricks. And here's how this works. So it's kind of like a blend between some microarray ideas and DNA sequencing ideas. And so here's how this works. The most important, one important aspect of this is the following. So you know, based on microarrays, you can take mRNA and turn it into cDNA with reverse transcriptase. And so you get this pool of cDNA. What you can actually do then is do something that seems a little crazy. You put it in a sonicator and fragment it all up into tiny little fragments, maybe around 50 to 100 bases long. So you can sequence the genome, or you can sequence the transcriptome. The genome would be just sequencing all the DNA you get out of a cell. The transcriptome is sequencing all the cDNA. But the trick is, it's kind of like a backwards microarray. You take all of the, let's just think about DNA for now. You're sequencing the genome from a patient. You want to sequence a whole patient's genome. You take all their DNA and you fragment it into 50 or 100 base pair fragments, and then you randomly affix each of those fragments to one place on a grid, kind of like a microarray, okay? And the neat thing is you can attach millions of fragments to one of these chips, okay? And then what you do is you're going to use fluorescence to identify where the DNA is adhered, and this is actually called a flow cell. And then you do a miniature little sequencing reaction on each DNA strand that has been adhered to this solid array. And the way this is done is kind of interesting. It turns out that you can attach something called a primer or a linker to the end of all of these random different sequences of DNA, and then you can get an oligo that you know hybridizes to that. So all these immobilized pieces of DNA have been modified to have the same linker on the end, and then you can come along with a primer that is complementary to that, and you start sequencing the little stretch of unique DNA, which is shown in gray here. And the way you do that is you keep adding fluorescently labeled nucleotides in 4 colors corresponding to each base. And so you go through cycles of this. So here's the first cycle, and this is just zooming in on a little region of one of these flow cells with maybe 15 different spots of DNA, and we'll follow a couple of them here. Here's one that is, I guess that's green right there, right? So the first cycle, that spot is green, and you know, this one over here is blue, that one's yellow. There's some nucleotide for each one, for the first nucleotide added. And then you wash it and do some chemistry, and then you add the next one and the next one. So you follow this single spot, and you can see that it goes like green, blue, red, green, yellow, red. And that corresponds to the sequence, which if you look at the little code of how they're labeled, it ends up being CAT, CGT. But what you're actually doing this is you're doing this in parallel for millions of DNA fragments all at the same time, okay? And so you can sequence the entire human genome, which contains 3 megabases of DNA, sequencing all these little fragments at the same time. And then you get a lot of data. that you have to make sense of, but it's a very powerful parallel mass sequencing approach. So how does this work? It turns out that the chemistry of the nucleotides, again, that's very important. So these nucleotides used in next generation sequencing are called cyclic reversible terminators. And the chemistry kind of shows you how it works. So here are two examples. They both function in similar ways. But what you'll see is that it has an interesting chemistry at the 3' where the hydroxyl is supposed to be. So that's actually blocked, and it can add to, first thing you do is you basically add this to the growing strand, and we'll add this, and then you can see what base it was by the color of the fluor that's attached. So you need a combination of 4 different ones, each corresponding to each base, right? So this polymerizes, but it's blocked because there's interesting chemistry at the 3' region of the deoxyribose. And you can read what base it was based on the color. And then it's called a flow cell because you can flow different solutions over this. The next thing you do after that is you put in a chemical buffer that actually cleaves this bond here, so you cleave off the color, okay? And then you wash it, and then the next solution converts this business into a hydroxyl group. So now it can accept another nucleotide. And then you go through the second cycle. And so each successive cycle is polymerization, reading the color, cleaving off the color, and then deprotecting the 3' hydroxyl group, turning it into a hydroxyl. And then you just repeat that process over and over. So you can actually end up getting all this sequencing data. But then you have to make sense of it, so there's a lot of computation involved. And there are a lot of variations. So one particular technology is called RNA-seq, and that's where you're using this next-gen sequencing to sequence RNA, actually cDNA. And what you can end up doing is you get all of this data, and then you have a million different 50-base sequences, but 50 bases is a unique sequence in the human genome, and the computer matches up where that sequence should be in the human genome. So you align all those sequence results, sequence reads, to the reference genome, which is the human genome. And here we're just looking at a portion of the human genome, and we can see this pattern in tissue 1. Each one of these is counting up where you got a sequencing reaction. And here's tissue 2. So obviously there's a lot more sequence reads in tissue 2, but you get this interesting pattern like this. Why do we get that? It turns out that those are the exons in a particular mRNA, and the places that didn't hybridize are the introns. The reason you didn't find them in the mRNA is they get spliced out and thrown away, okay? And so this is an example of, we can tell that this particular gene, this particular mRNA is more expressed in tissue 2 than tissue 1, and we can quantitatively say, hey, it's like 10 times more expressed, okay? Because we're counting up the number of sequence reads in that mRNA. But you always get out only the exons, not the introns, because of splicing, intron removal, okay? And so computers can do this for all the messenger RNAs in the cell at once, okay? And you can get gene expression changes like this. And it's far superior to the old microarray approaches. And this is how all gene expression is assayed these days. There are other tricks you can do this. So that's RNA-seq. You can also do ChIP-seq. So ChIP stands for chromatin IP. You remember that chromatin contains modifications, can be acetylations of histones, methylations of histones, stuff like that. In this case, what you do is you take the whole genome, and you isolate chromatin, which is the complex between DNA and protein, and then you fragment it up into like mononucleosomes, and then you use an antibody that's specific for a particular post-translational modification, or even where a protein sticks, like p53 to DNA. But you can immunoselect all of the chromatin fragments that contain a particular modification or a particular protein stuck to it, okay? And then you throw away the rest of the DNA or the chromatin that doesn't associate with that antibody. So the antibody is the trick here. You can immunoselect all chromatin with a particular modification. And then what you do is you add protease to the tube. You dissolve all, or you just destroy all the protein, and you're left with just the DNA. And then you can do next generation sequencing on the remaining DNA. And then you can align it to the genome, find out where those sequence reads match up, and you can figure out the distribution of, for instance, an antibody, in this case p53, across the human genome that way. So we can map out chromatin like this too using ChIP-seq. So you've seen RNA-seq, ChIP-seq. If you're just sequencing DNA, that's DNA-seq. And yeah, pretty interesting method. So that's a kind of a real short course on next generation sequencing, but now you know how it works. Very powerful method. So now let's shift gears and look at how you would use this for real experiments. So we talked about nuclear receptors on Wednesday. And here's our friend, the estrogen receptor with the DNA binding domain and the ligand binding domain. Since it's estrogen receptor, it binds estrogen, or it could bind estrogen-like drugs, like raloxifene, for instance. And so you now know that these type 2 nuclear receptors, the ligand diffuses through the membrane, causes the nuclear receptor to dissociate from HSP90, and it dimerizes, goes into the nucleus, and helps to activate transcription, right? That's how type 2 nuclear receptors work. So this is an example of total global analysis of messenger RNA expression using a heat map. And what we have here is a whole bunch of different drug treatments on the same kind of cells. So we have a total of several different drug treatments using various estrogen receptor antagonists on a single cell type, and then we've clustered it into one of these heat maps. And we can see how each individual, and here's a control at the top, we can see how each individual treatment of one or two drugs at the same time affects gene expression in these cells. These are probably, they may well be breast cancer cell lines, I think. And so we can see how different cancer drugs affect a given cell type using different combinations of drugs. And the red means it's activated. And then you can, so this hierarchical heat map clustering really helps you figure out which genes turn on and which genes turn off. You can then develop these Venn diagrams and look at the different genes that are turned on and turned off in these different drug treatments. So we got fulvestrant, raloxifene, tamoxifen, acolibeine. I'm not as familiar with that one. But at any rate, 15 genes were activated by all these treatments. And these are, we can also identify the function of the genes. The ones contained in that cell are all cancer related. So we can see that all these drugs activate 15 genes. We can look up what they are. But here's one that's activated by, here's one that isn't touched by any of the drugs. These three over here are uniquely activated by raloxifene. You can really make sense of this. And so you can kind of plow through all the data and make sense of what's going on. And this just shows the different number of genes activated by each of the drugs. So you can extract all this transcriptional data that tells you how drug treatment is affecting transcription, mRNA content in cells. And you can furthermore distinguish between upregulated and downregulated genes. So for instance, the drug fulvestrant activates a whole bunch of genes, but it doesn't really downregulate many genes. Tamoxifen activates quite a few genes, but doesn't silence many genes. This one over here activates a fair number of genes, and also turns off a number of genes. Here's raloxifene, very similar to tamoxifen, but more gene activation. So you can really compare how different drugs work. And you can always dive down and find the identity of any of these genes because you know their sequences and mRNAs. So that's GPCRs. Now let's look at transcription factors, or transcriptional, I'm sorry, that was nuclear receptor stuff. Now GPCRs, G protein coupled receptors. Let's look at an example there. So here is epinephrine, and it's not really showing the GPCR, it's just kind of indicating that you have a G protein. It must be a GS because it's stimulating adenylate cyclase to make cyclic AMP, and then that is initiating phosphorylation of a kinase cascade, which ultimately then, the last kinase is actually regulating conversion or mobilization of glycogen and metabolism into glucose, okay? So epinephrine causes all this signaling, and then ultimately you get glucose, okay? But there are other GPCRs that actually, so this is kind of like non-transcriptional. Well, this is a non-transcriptional response, but there are other GPCRs that actually have signaling pathways that end up activating genes. And so this is not a pharmacology example, but it shows you something like this. So this is RNA-seq analysis of different types of blood stem cells, hematopoietic stem cells. And, I won't go into all of this, except to say that what we're looking at here is activate, a specific GPCR has been activated here, and then we're looking at different samples of cells, endothelial cells, hemogenic endothelial cells, hematopoietic stem cells, different types of cells. And you can see that this G protein is regulating a number of genes, but you can also see, interestingly enough, that the same GPCR is causing different transcriptional responses in different cell types, okay? And that's because they're wired up differently with their signaling pathways internally. And so this is kind of an interesting example because signal transduction cascades can be incredibly complex, and we don't have them all figured out. But we can do tricks with the genetics to find out, even though we don't know all the internal signaling pathways, we can find out what the end result of a receptor's function is by simply assaying changes in gene expression. And you don't have to have any knowledge of what goes on in between the receptor and the output. So that's pretty powerful because it's hard to figure out all these signaling pathways. Okay, so this sort of stuff is done with most drugs in development. We want to know what kind of genes turn on and off with any drug treatment. And it can be really informative because we know a lot about gene function. It's been really important in cancer treatments because cancer is sort of a genetic disease, and we really want to know how drugs are affecting cancer genes. And these types of studies allow you to assess relative activities of different drugs by looking at their transcriptional responses. Okay, that's a lot of genetics. There is going to be a whole seminar on this, I think on December 4th. Rachel O'Neill, MCB professor, which will probably be in the next room over there. So come catch that, and you'll see more of this sort of stuff if you're interested in next generation genomics. Okay, but there's every single drug company, pharmaceutical company is doing this kind of stuff on their drugs in development. And it's a great discovery tool as well. All right, so the last thing I want to do today is give you a quick overview of what we've talked about, just hitting some of the major points, and then we'll shift on to some other things next week. We're going to talk about ion channels and transporters and the drugs thereby, but I want to kind of summarize things thus far. And so this is stuff that's all been on the, going back to dose response stuff. But, definitely just a bit of review here. Remember occupancy theory, know all these, what EC50 is, Emax, TD50, LD50, know how to interpret dose response curves, know the differences between individual biologic dose responses versus population-based dose responses, therapeutic window, therapeutic index. Definitely know the difference between potency and efficacy. And for GPCRs, know about the structure of GPCRs, how many GPCRs there are, just be aware that there are a lot of them. Know that they undergo conformational changes when ligands bind. Be aware of G proteins also, that there are all these different types of G proteins out there. Obviously, the G alpha subunits are really important, right? And the beta gamma is also important. And you get different signaling pathways by G alpha and beta gamma. Know that G alpha is activated by GDP gets kicked out, GTP becomes associated, and that leads to downstream signaling. The G proteins affect second messenger signals and produce second messengers, things like cyclic AMP, IP3, and that these second messengers, once produced, oftentimes trigger kinase cascades, phosphorylation cascades. But ultimately, the last kinase has to activate some enzyme that's going to do something, right? Or it might do something like cause a transcription factor, translocate into the nucleus, these types of things. But don't forget that the beta gammas kind of tend to stay as a dimer, but they also participate in signaling pathways. GTP activating proteins and GEFs, GTP exchange factors, these regulate signaling as well. This should say G alpha, but there are 20 different proteins that are G alphas, but they can fall into these four families: GI, GS, GQ, G12, 13. We talked a lot about GI and GS and GQ. This is more of a specialized one down here. But remember that each receptor, each individual GPCR, is going to be associated with one and only one kind of G alpha protein. So some receptors might stimulate the production of cyclic AMP, others might inhibit it, depending on how that receptor G protein complex is configured. This is a good kind of review thing as well, knowing that basically G proteins, it's just a good diagram of how GPCRs work. Knowing that the alpha subunits, alpha S is quite different than alpha I. This one stimulates adenylate cyclase. This one inhibits it. Here is an alpha Q version that activates phospholipase C, making the second messenger IP3 and diacylglycerol. So basically just sort of review stuff. Know about drug targets. Know that GPCR drugs are very common. We also went through orthosteric and allosteric drugs, and that, these activate or inhibit downstream signaling pathways, signal transduction cascades, oftentimes phosphorylation. The responses can be non-transcriptional, like mobilizing glucose stores, or it can be transcriptional, like what we were talking about today. You can get really quick responses, like if channel opens, or you can get really delayed responses, like you affect the expression of a bunch of genes. Yeah, nuclear receptors, the distinction between class 1 and class 2. The class 1s are the ones that can translocate into the nucleus from the cytoplasm. Class 2 are obligately in the nucleus. Know about heat shock proteins, Know that most of them dimerize, but some of them heterodimerize for activity. And that dimerization for these class 1s is always associated with the ability to translocate into the nucleus. And of course, know that they're transcription factors as well. So nuclear receptors are both receptors and transcription factors in one protein. Today, also just kind of know that there are these great next-gen sequencing techniques to assess transcriptional responses, etc. And know a little bit about how transcriptional responses mediate cellular responses. So if you transcribe a whole bunch of genes involved in driving the cell cycle, that's going to make the cells divide, right? That kind of a thing. Okay, so anyway, that's kind of like I guess we're a little bit early today. It's fine. It's Friday right before Thanksgiving break. But I want to go through a few things so that you can kind of cement this in and don't eat so much food that you forget this stuff. Okay. But when we come back, we're going to switch over to one last section next week. And like I said, we're going to talk about ion channels, transporters. Kind of we'll deal with both of those separately. And then on the final Friday of the course, we're going to talk about the drugs that affect ion channels and transporters. So that's where we are. Everybody have a good Thanksgiving break. Enjoy your extra 9 minutes. And by the way, there will be an exam review on the 4th floor atrium if you want to see what you got wrong. Coming right up. Okay, in a few minutes. We'll start that off in like by 11 o'clock or so, okay?